So today, I'm going to talk about Ribonanza, deep learning of RNA structure through dual crowdsourcing. Before I get started, I want to give a little bit of a background on how I got into RNA. So like Joe mentioned a few years ago, I participated in the Open Vaccine Challenge, um, and actually placed pretty high. And as a result, I actually co-authored the paper with Riju and everybody at DASLab and a bunch of other Kagglers. And after that, I kind of had the idea to go into sort of some research with mRNA stabilization. And since then, I have actually become a Kaggle Grandmaster. And actually, wrote some grants that got a lot of funding. And actually also published my results in Open Vaccine as a separate paper with some more detailed technical insights. And about July last year, Riju reached out to me about potentially another open vaccine, but with 100 times the data. And the main objective of the competition was to help us achieve the dream of RNA structure modeling, which is just going from an RNA sequence directly from a 3D RNA structure. And it sounded very enticing, so I was just like, okay, let's do it. And a main bottleneck of RNA structure modeling is basically with data. Because currently we only have about 1,000 high-resolution 3D structures on the protein data bank. But in order to solve this data bottleneck, we turn to a different type of data, which is basically chemical mapping data that uses readout of unpaired nucleotides to essentially attack RNA molecules with a chemical modifier and read out the changes in the sequence. And as a result, we can get basically a 1D signal along the RNA sequence dimension that tells us how reactive each position is. So this signal basically looks like this. So you get some reactivity value at each nucleotide position, and this is supposedly highly correlated with RNA structure. So although we kind of get a good signal from the chemical mapping experiment, it's still a one-dimensional signal. When you try to predict RNA secondary structure, you need to kind of predict sort of, the pairwise interaction, which is basically what base pairs are forming hydrogen bonds with different base pairs, which is kind of like a 2D prediction problem. And a way to actually use chemical mapping data or chemical mapping experiments to probe the secondary structure is actually with something called mutate and map, where you go along the sequence dimension and change each nucleotide to its complementary part one at a time. And you can basically get a 2D profile where upon mutation, some of the positions will have increased reactivity. If you see on the diagonal here, you see some black spots, which indicate increased reactivity, and essentially that these nucleotides are probably paired before the mutation. And the actual key result of this experiment is you'll see off-diagonal features that are indicative of sort of the interaction between base pairs. So basically, you can then use something like M2Net, which is basically a very small convolutional neural network to convolve on the 2D chemical mapping profile to get a pretty accurate secondary structure prediction. And you see in the red and purple circles, these are basically secondary structure formation that are shown by the mutant map experiments. So with this, we kind of know that we can probably learn secondary structure from this type of data. And on the right hand side, you can see the actual secondary structure mapped out using these mutant map experiments. So now, if we have an oracle that can accurately predict chemical mapping, you would have to understand RNA structure, and in order to do that, we first need data. So question is, how do we get 1 million diverse sequences? And the next is we actually need people who do machine learning to train the oracle, which is basically a machine learning model. And it turned out the way to do that is by dual crowdsourcing. So we will combine these two different platforms. The first one is Eterna, where folks design intricate RNA structures, and then Kaggle, which has been a staple platform for comparative machine learning, where people build very advanced and accurate machine learning models. And as I mentioned before, this actually also has been done a couple of years ago in the open vaccine competition, where Eterna players provided diverse sequences for training, and Kaggler's trained machine learning models that could accurately predict RNA degradation. So fast forward to the Ribonanza competition, we wanted to create a model that can predict structures of any RNA molecule. And to do this, we need data. So at the beginning of the competition, we were able to have about 1.1 million RNA sequences, and each of them have two profiles, namely 2A3 and DMS. And in order to ensure a rigorous testing process, we actually measured the 1 million RNA sequences in the test set during the competition. So even we as hosts did not know the profiles of the test set sequences. And very importantly, because we wanted to test the generalization abilities of top models, we actually included longer molecules in the test set than in the training set. And from a machine learning standpoint, it's basic. The task is basically a sequence prediction task, where comparators are asked to predict 2A3 and DMS reactivity and each nucleotide. And we decided to use mean absolute error as the metric to evaluate models. All right, so first, I kind of want to show a little bit about the power of Kaggle community. So within a few days, actually, IFOS, whose real name is Maxime, he actually put up a notebook that scores really well using just the transformer. So this was really good because the base it's kind of a pretty strong baseline, but it's not strong enough where everybody just ended up using the same model. And he also created sort of a submission notebook that does inference. So it's basically a really, really good starter kit for the Ribonanza competition. And that's, I would like to talk a little bit about the transformer. So what it is and what it actually does. So The transformer was first introduced in 2017 paper called "Attention is All You Need," and it was first used for machine translation tasks, but later on it has been kind of adopted to do many other things, such as protein folding and playing StarCraft, and even image recognition. So the core mechanism behind the transformer is called the attention. And in our case, we're using something called self-attention, which is basically using sort of a learned soft graph structure to aggregate over items in the sequence. So if you think about the RNA sequence and how different base pairs interact with each other, you would think that you would want to kind of get some information from the other nucleotides when you're modeling each nucleotide. So that's kind of what the transformer does. It just looks at, for each nucleotide, it looks at all the other nucleotides and you get some information from them. So during the competition, we actually realized that some of the comparators were probably doing something wrong with the modeling process, because we looked at some of the mutant map predictions of the early submissions, actually one of the top early submissions, and you can see that this one, it doesn't really show any off-diagonal features that are indicative of secondary structure, and it kind of just looks pretty strange. So at the time, we were actually kind of worried that because in the public test set, where people can actually see how well the model is doing, there weren't any long sequences. So we were worried that people didn't have a way to do sanity checks, and they didn't understand the importance of model performance on longer sequences. So we decided to actually make a post on how to do sanity checks on if the model generalizes to longer sequences. So what we ended up doing is we took a really long sequence that actually whose actually M2 sequences were part of the test set, and I used one of my models, which gave pretty reasonable M2 predictions that are pretty correlated with this actual secondary structure of this sequence. And this is actually shown on the top right, the sequence. And we actually had some pretty good discussions with comparators where people started showing kind of their predictions on this model. And I think that was like a really good way for us to kind of guide comparators to make sure, they're not kind of ignoring this important part of the game, which is how the model generalizes long sequences. And during the competition, I was also doing a lot of testing, actually with the RNA deck format, which I created for the open vaccine competition. And on the left-hand side, you can see kind of a diagram of how the model works. So the RNA deck format is essentially a two-track model that has a sequence track that basically operates on a sequence representation with a combination of 1D convolution and multi-edit self-attention. Essentially a transformer with some convolutions. And on the pair track, because in open vaccine, we were provided with base pair probability matrices, which are basically some pairwise representation of RNA structure that was predicted by conventional RNA folding algorithms. I decided to use actually 2D convolutions to operate on that, and then add that as an attention bias into the self-attention function as a way to influence how the model uses self-attention. So this was helpful because I think in open vaccine, because we only had 2.5K data, it was not possible to learn really accurate secondary structure. So, and we actually did scaling tests where we kind of try to figure out when we can actually get away with not using this sort of prior information, which is base pair probability predictions as an input. So in this case, it seems like if I remove the base pair probability matrix as an input for the RNA deck format, it doesn't look like it, at the current scale, it was really surpassing the model that has this prior information as an input. But it looks like as we scale up, at some point, you will be better to use a model that only takes sequence as an input, and probably has learned a more accurate representation than the base pair probability predictions by actually, in this case, Eterna. And we actually also looked at how much data we need to get good mutant map predictions. So in the first row are predictions from RNA deck format with base pair probability matrices as inputs. And very interestingly, even at 1.4K sequences, it does pretty reasonably actually on the mutant map sequences, probably due to Eterna being okay at this as well. And as we scale up to more sequences, it becomes a little bit better. And in the second row, we actually see something more interesting, where at 1.4K sequences, there's really no response to any mutations in any off-diagonal positions, which kind of means that the model probably just has no idea about RNA structure. But as we get to 14K sequences, we see that these off-diagonal features start to emerge. And this kind of shows that really 14K is probably the lower limit for a model that doesn't require any prior information to learn RNA structure. And then moving on to 140K sequences, the model starts to do even better. And following the competition, after our testing, we got to see what the top models were doing. And it turned out RNA deck format still did pretty well. It was actually placed in the top 20 as a benchmark. But some of the other top models are essentially also non-vanilla transformers. So the first place model is a transform model with dynamic positional encoding plus convolutional neural network. So this is actually in spirit very similar to the RNA deck format, and also the comparators acknowledge that they took inspiration from my solution, Open Vaccine. But moving on to the third place solution, we actually see something a lot more interesting, because it's a model that only requires the RNA sequence as an input and does not really need predictions from Eterna to do well. And how he seemed to achieve this was first using a different type of operation on the pairwise representation, which is borrowed from AlphaFold. I'll discuss this a little bit later as well, but this is called triangle update. And another key difference is that in the RNA deck format model, and the top solutions kind of inspired by RNA deck format, information only flows from the pairwise representation to the sequence representation via the self-retention function. Whereas in the third place AlphaFold style model, sequence representation, there's information flow from the sequence representation to the pairwise representation via something called outer product mean, which is essentially a pairwise outer products of the sequence representation into and added to the pairwise representation updated by triangle updates. So this just means that in the latter formulation, this information flow both from the sequence representation to the pairwise representation and the other way around. All right. So, an important distinction between some of the top models, I guess difference between some of the top models is how they operate on the pairwise representation. So some of the models that use Bayesian probability as the input does 2D convolution, whereas the models that only take sequence as the input use triangular updates. So first, for the 2D convolution models, they all rely on Eterna fold to have some sort of prior information on the secondary structure, and they're quadratic complexity in space and time. And then moving on to the triangular multiplicative updates, these actually produce pairwise representation completely from scratch. And it kind of involves looking at basically triangles formed between the ith node and the jth node and some other node, and looking at all the triangles, where you iterate k from left to right, and creating a new representation in the IJ slot. So I guess from a technical standpoint, a key difference between this and 2D convolution is that for the new IJ position, you can actually see all the other nodes in the graph, whereas for something like 2D convolution, you can only see other nodes that are close to either I or J. So if you imagine complicated 3D structures, a lot of times there might be more than just two nodes that are close in position interacting. So I think that kind of encodes some of the 3D structure modeling bias that we might want for some of the downstream stuff. And this one is cubic in time, because you have to go through, every row for each of the IJU connection in the output, which is a square. But luckily, this is quadratic in space, so it was not a big memory bottleneck. All right. So during the competition, we also kind of looked at sort of how using the Bayesian probability matrices predicted by these conventional algorithms, which do not really predict pseudoknots, how that fact impacts the model. And essentially what we saw was that when we train a model with only the sequence as an input, the model can learn these pseudoknots circled in red, in some cases really well when we look at the Newton map predictions. Whereas for a model that has the inductive bias from the Bayesian probability matrices, it tends to not really react to these pseudoknots, despite the fact that a model that uses the Bayesian probability matrix has actually a little bit better MAE. So this kind of shows that using the Bayesian probability matrix is maybe more of a limitation and a crutch than something we really want to do when we create something like a really good model. An important question is obviously the models learn RNA structure, and we kind of looked at some more kind of like individual cases. So this one was coronavirus frame shift stimulation element. When we looked at the top models such as RNA deformer, we see some pretty well-defined off-diagonal features, which are actually in pretty good agreement with other top models. And then when we looked at the experimental measurements, they're also in line with predictions from the top models quantitatively. So this kind of means that top models have learned a pretty accurate representation of secondary structure that can also be probed by mutant map experiments in silico. So this is kind of nice. So it looks like we're getting close to the to having an oracle, right? And yeah, here's a representation of the secondary structure. So unfortunately, when we moved on to some of the longer sequences, such as tetraamino ribozyme, it seems that the top models haven't really kind of converged on this sequence yet. So in the B panel, you can see the reference secondary structure that was extracted from the 3D cryo-EM 3D structure. And in many cases, there are a lot of wrong stems being predicted. And sometimes some of the important secondary structure will actually be missed. So this kind of shows that, you know, the models actually haven't completely converged on some of the more difficult sequences. All right, and before moving on further, I actually want to discuss the metric really quickly. So was mean absolute error the right metric? And so how we kind of arrived at using MAE was because we did some simulations with the submissions from Open Vaccine, where we try to maximize the Spearman rank R, which is basically how well the public ranks and the private ranks correlate with each other. And we tested a bunch of metrics that seem like they would be reasonable for this competition. And what we found was that from using data from Open Vaccine, mean absolute error was actually the best metric in terms of preserving the ranks between public and private. And fast forward to the end of Ribonanza, we did the same analysis, and we found out that MAE was still the best. So this kind of shows that MAE was probably the right metric for this. All right, so now I want to talk about the distillation, of the insights from Ribonanza into a single model for downstream tasks, such as our secondary structure. So we really wanted to do this not only from a modeling standpoint, but also from a data standpoint. So because the top models in Ribonanza were fairly accurate, we thought it might be a good idea to actually get the predictions from the top models and use them as pseudo labels to do semi-supervised learning. And we also want a model that can easily make 2D predictions. So basically, a vanilla transformer really struggles with 2D predictions because it only has sequence representation. You can do something like using the self-attention weights as features to make 2D predictions, but it really just doesn't work very well. And the reason for this is that many RNA tasks require 2D predictions, such as basically secondary structure predictions or distogram predictions, angiogram predictions. So another thing is that in these machine learning competitions, usually the top models end up being some huge ensemble of 10, 20, 30 different models, which can be really inconvenient if a biologist wants to use the model. So so to kind of solve that problem, we wanted to have just one model with good performance without assembling. And we also want obviously the model to be easy to use. So it should just work for downstream tasks. And then we basically created RibonanzaNet, which is basically something that fulfilled all the considerations we had in mind when we were thinking about how to design something like this. So firstly, this model makes predictions directly from sequence without needing base pair probability matrix. So this removes the need of using something like return fold and also having that installed on your machine. And it's also immune from some of the problems some of the model had earlier with pseudoknots, where the model would just very conservative on predicting pseudoknots because the inductive bias from the conventional algorithms. Sorry. And this model kind of takes a lot of inspiration from the third place solution and evil former. And it seemed like 1D convolution was still necessary, which was slightly surprising considering AlphaFold doesn't use 1D convolution. And very importantly, this model has a fully learned pairwise representation with global updates. So this model can easily make 2D predictions, unlike some of the other top models who don't have really like a good pairwise representation. And structurally, this model is pretty similar to our deck format. Actually, it has two tracks, one track to process the sequence representation, and another track to process the pairwise representation with triangular updates. Very importantly, in this model, information flows both from the pairwise representation to the sequence representation, and from the sequence representation to the pairwise representation. And in the end, we kind of use about 9 blocks of these consecutively. And this was trained on basically reactivity data. And an important thing to consider is how we use predictions from top solutions. So we realized actually most sequences in Ribonanza are low SNR, meaning that most sequences don't actually get a lot of reads in sequencing, which means that usually these were not really used during training. And what we could do is we could create pseudo labels from the 1.9 million sequences that were relatively low SNR, which ended up being actually 10 times of high SNR training sequences. So what we tried first was actually just combining the pseudo labels of noisy training data from the top 3 teams. And we pre-trained RibonanzaNet with these pseudo labels. And this was already on par actually with top solution, which is despite being just one model, and top solution being an ensemble of 28 models. And then as we move on to including even sequences from test data as pseudo labels, and pre-training RibonanzaNet with those pseudo labels, we see that RibonanzaNet actually surpassing the top solution while being just a single model. So next, we try to fine-tune RibonanzaNet to do some downstream tasks. So the first of which is basically predicting sequences that drop out of chemical mapping experiments. So this was done by Hamish. So actually in Ribonanza, most of the data is kind of low SNR, so we get about 20%. of data, so that's actually usable. So what if in the future we can know beforehand which sequences are going to not get a lot of reads, are going to be low SNR in experiments, and we just don't really synthesize them and do experiments on them. And then we can only synthesize sequences that are going to be high SNR as a way to increase the yield of our experiments. So one way to do that is if we can predict which sequences are going to be high SNR, we can just only keep the sequences that are high SNR. And this was seemed like this could be done with RibonanzaNet, despite the fact that the primary objective of RibonanzaNet was probably to learn RNA structure, but it seemed like it was also able to learn what kind of caused some of the sequences to drop out. And in this graph, you can actually see two examples of sequences that are that have the same actually secondary structure, but vastly different number of reads. This kind of just shows that RibonanzaNet has learned not only RNA structure, but perhaps some other insights about these chemical mapping experiments that allowed it to predict sequencing reads. And next is actually a revisit to open vaccine, where we try to fine-tune RibonanzaNet to predict RNA degradation rates. So this could be pretty important for designing stable mRNA vaccines. And what we see is that RibonanzaNet vastly outperforming other top models in open vaccine, and even outperforms post-competition model, which was trained by me, basically the semi-supervised RNA deformer, that was an ensemble of 50 models, each of them test time augmented with 12 secondary structure predictions from different folding algorithms. So this is basically, the orange is basically 600 forward passes per sequence to make one prediction, whereas RibonanzaNet is just one model. That's just one forward pass. And RibonanzaNet actually does better. So this really shows kind of the power of a strong pre-trained model, and how it can be fine-tuned to be very powerful in some of these downstream tasks. And another sanity check that we did was actually sort of like a zero-shot test of predicting mRNA half-life. And how this works is that with a model that predicts RNA degradation rates, if we sum up the degradation rates in each nucleotide position, that sum should be correlated with half-life. And I actually did this test with my model from Open Vaccine. And with this data set, we actually had sequences that were up to 1,200 nucleotides. So it's really a good test because, you never know if a model trained on RibonanzaNet data, which is primarily 200 long, you never know if it's just going to fail on some of these long sequences. And it turned out that it does pretty well on the mRNA half-life prediction. When you look at the prediction, the correlation of the sum of degradation rates, it sometimes outperformed RNA deformant, which was kind of previously the best. And it also does really well against, something like Vienna, which despite doing well on the nanonuc sequences, it seems to fail sometimes on some of these other mRNA sequences, such as MEV and GFP. All right. So next, I'm going to talk about kind of fine-tuning RibonanzaNet. So previously, we have done a lot of probing with mutant map experiments, where we basically made predictions for all the single mutants of a particular RNA sequence, and then we can look at the mutant map profile to see sort of what the model thinks the secondary structure is. Although it was a nice test, this was basically not a direct prediction of secondary structure, and it was not that easy to directly get a secondary structure prediction. And sometimes when you use something like M2Net, which is previously developed to basically get a secondary structure prediction from butane map profiles, sometimes it doesn't do that well. And however, when we fine-tune RibonanzaNet to predict RNA secondary structure by using basically about 800 secondary structures extracted from actual 3D structures on the protein data bank, we saw that RibonanzaNet was able to produce really accurate base pairs that are also visually in line with the profiles predicted on the mutant map experiments. And after using sort of the Hungarian algorithm, we kind of get a really nice secondary structure prediction. And on this one, actually, we also kind of figured out a way for the model to kind of predict how well it's going to do on a particular sequence by just looking at the confidence of the predictions. So for this one, the model seems to be pretty confident. It gets EF1 0.86 and EF1 CASP of 0.8. I should know the EF1 CASP is basically an F1 score on pseudoknots. Then we looked at the sequence that really caused a lot of problems with, in the test set, which is the tetramer ribozyme sequence, and we looked at how well the secondary structure predictions correlate with the mutant map experiment predictions. We see that the model does output something pretty consistent with the mutant map profile. So this kind of shows that whatever the model learned from chemical mapping data is actually being, translated very well into, base pairs after fine tuning. And however, compared to the ground truth secondary structure, the RibonanzaNet is still getting a lot of actually some of the secondary structure wrong. So it kind of shows that there's still work to be done, especially on these longer sequences. And this one gets EF1 score of 0.8, and notably an EF1 CASP score of only 0.46, which is pretty low. And that kind of just shows that the model is pretty confident about overall secondary structure prediction, but not so confident about basically the pseudoknots, which kind of makes sense because usually pseudoknots are more difficult to predict. And then we kind of did a more comprehensive test against basically the top secondary structure prediction algorithms that are currently available. So we see that on the PDB set, RibonanzaNet is certainly the best, followed by actually Shapefied HFO, which I think is an unpublished algorithm, then by Viennafold. And interestingly, on the CASP 15 secondary structure test, RibonanzaNet is doing vastly better than the other algorithms, as is shown, and pretty obvious on this plot. So the predictions from RibonanzaNet is very tight, around 0.9, over 0.9 actually, the median. Whereas the other algorithms really seem to struggle on these sort of blind tests compared to RibonanzaNet. So these tests kind of just shows that RibonanzaNet really is state of the art in RNA secondary structure prediction. All right. So the next task is actually we wanted to test, if just inputting the correct secondary structure or an improved secondary structure prediction as input to an existing 3D model that actually uses secondary structure predictions as an input and evaluating kind of, if just doing that will give us better predictions. So we kind of looked at, this sequence, both using sort of RibonanzaNet secondary structure prediction and also SPA-RNA secondary structure prediction, which was the default model for TR Rosetta RNA to predict secondary structure. And we do see kind of a noticeable improvement in RMSD, so on the left, the predictions from RibonanzaNet secondary structure has an RMSD of below 5, whereas on the right, the 3D predictions using SPA-RNA secondary structure has RMSD of over 10. So this shows that we may be able to just get better 3D models by using basically better secondary structure predictions from RibonanzaNet. And here's another experiment where we did where we kind of inserted more base pairs in this GU kissing loop sequence, which I'm sure the Eterna players probably know this sequence. So essentially what we kind of did was, we inserted either 5 or 15 base pairs in between I think these kissing loops. And what we kind of thought would happen was that as we insert more, if the model really understands 3D, it should stop to predict this kissing loop structure. Unfortunately, that doesn't happen here. But what we do see is that the model, as we insert more base pairs, the model becomes less and less confident. So on the pseudonode. So moving from the first plot to the second one, you'll see that the cross-pair EF1 going down from 0.79 to 0.71, whereas in the last one, where we inserted a lot more base pairs, the cross-pair EF1 score goes down drastically to actually 0.4, showing that model becomes much more uncertain about this GU kissing loop. So this kind of shows that the model may understand 3D to some extent, but not enough to stop predicting the GU kissing loop pseudonode. And another reason why this happened was maybe because the threshold from the Hungarian algorithm was not set high enough. So it could just be a technical reason, but I think the model does understand 3D to some extent. All right, so here's, I guess, another application of RibonanzaNet, which is doing a genome scan to discover interesting stool-knotted structures, which is actually done to get sequences for RibonanzaNet 2. So we kind of created this ensemble score called open knot EF1, which is basically an average of the global EF1 and the cross-pair EF1. So this is basically inspired by the open knot score, which was used to create some of the stool-knotted designs in RibonanzaNet. And how this kind of works is just you scan the genome with RibonanzaNet, calculate open knot EF1, and then you can sort these sequences based on the EF1 score And actually also added a waiting term determined by the number of pair positions, because we kind of wanted sequences that are well structured. So doing this can basically help us achieve that. And we actually already found some very interesting structures. So this is one of the top sequences from the genome scan, which is basically highly structured and forms a SONA where you can see the strand actually goes through the loop right here. So this one gets open knot EFR of 0.88 and 160 out of 200 nucleotides are paired. All right. So finally, I guess all the notebooks and data are available on Kaggle and GitHub. And actually, I created this notebook on Kaggle where you can fine-tune RibonanzaNet to get state-of-the-art assembly structure prediction performance in under 10 minutes. So you can see the runtime right here is 550 seconds, which is pretty great. So, and obviously, RibonanzaNet training code is available and training data. Training code is, probably too expensive for the average person to run. But, if you're interested, I think it would be interesting to look at. All right. And I'd like to thank everybody at the DOS Lab. And thanks to Riju for inviting me to work on this. It was really fun. And also, the top Kagglers who provided us with really great solutions and ideas on how to kind of create like a really good single model in the end. And also all the funding sources, and all the folks at Kaggle, such as Walter and Maggie and Ashley. It's been pretty fun. And then, obviously the designers, RNA designers at Eterna, without whom really wouldn't be able to have these interesting sequences.